Unit 8, page 1. What is DNA and how does it relate to our genes? Let's think first. What do you already know about genes? Pause the video here and write down a couple ideas. When you're ready to begin, press play. Okay, so hopefully you've written some things about genes. Let's take a look. What is DNA? So, as we know, DNA is going to be found in our nucleus. So, although environment influences how an organism develops, meaning what's around them, the genetic information that is held in the molecules of DNA ultimately determines an organism's traits or characteristics. DNA achieves its control by determining the structure of proteins. Within the structure of DNA is the information for life, the complete instructions for manufacturing all the proteins for an organism. Remember we had learned about some proteins back in Unit 2? We had learned about some that make up enzymes, which are functional units of genes. Within our bodies, we're made up of trillions of cells, and within each cell is DNA. So bodies are made up of cells. All cells run on a set of instructions spelled out in DNA. So our body, and we can see here a picture of an animal cell, and then a picture of the DNA double helix. So you can see how DNA kind of looks like a twisted ladder. We're going to examine the actual colored spokes on that twisted ladder, so you can see the purple, the green, the yellow, and the red. We're going to take a closer look at those subunits of DNA. So parts of the nucleotide. DNA is a polymer, which just means that it's made up of smaller repeating subunits, called nucleotides, which are the monomer or single subunits. Remember that mono stands for one and poly stands for many. Nucleotides are going to have three basic parts. You have the sugar, which you can see here, deoxyribose. We have the phosphate group, which you can see denoted by the P, surrounded by these O's. This P stands for phosphorus, the O stands for oxygen. And then here we have the nitrogenous base. So the entire structure we call the nucleotide, but sometimes we'll talk about the bases of DNA, so we're talking about just this one piece. The structure of the nucleotide itself. In DNA, there are 4 possible nucleotides, each containing one of these 4 bases. The phosphate group and the deoxyribose molecule form the backbone of the chain, and the nitrogenous bases stick out like the teeth on a zipper. So if we look here, this is the sugar, the phosphate is attached to the very next DNA molecule. See that chemical bond? For the sugar, the phosphate. So the sugars and phosphates are that backbone, and then that base that we talked about before sticks out on the end here. Now the bases can be adenine, cytosine, thymine, or guanine. We just typically will say A, G, C, or T, but please remember that there are full names for each of these nitrogenous bases. The base pairing rules, so the structure of DNA. We're going to have certain bases matched together. A will always go with T, and G will always go with C. One easy way to remember this is that the curved lines, so to make the letter C it's a curve, and to make the letter G there's a curve, as opposed to write the letter A you use straight lines, and to write the letter T you use straight lines. But you should remember that T and A will pair together, and G and C will pair together. So you can see how DNA here is made up of 2 different strands, or 2 different sides. So we have here this left side and this right side. They both have that phosphate, sugar, phosphate, sugar. See how the sugar, phosphate, phosphate? And the only thing that's different are these nitrogenous bases in the middle. So again, it's a double helix, meaning that there's 2 sides. And it's kind of like a ladder. A pairs with T, and C pairs with G. So the structure of DNA, again, the 2 different sides are actually held together with a special type of bond called hydrogen bonding. We can see different ways that the DNA molecule is drawn, but they're all the same basic drawing. The structure of DNA is credited to two scientists, James Watson and Francis Crick, worked out the three-dimensional structure of DNA. They actually did this by creating cardboard cutouts that they fit together to figure out the shape, and you can see here their model of the DNA structure. But they couldn't have done this without the work based on two other scientists, Rosalind Franklin and Maurice Wilkins. And Rosalind Franklin is credited with taking these pictures of DNA, which allowed Watson and Crick to realize that it was this double helix. So she took pictures basically looking down the DNA molecule, so you can see that these would each be the bases.